It's really fun to participate. I'm just getting to know the synthetic biology community a little bit from my interactions with Ron Weiss around the Science and Technology Center funded by NSF at MIT that Roger Cam heads. And I was a little reluctant, actually, to get involved with that center initially because I looked, when Ron first came to MIT, I looked at what he did and I felt it's super interesting, but how is it relevant for the things I do, which is work in the field of tissue engineering. And as shown on this slide, the kinds of things we do in our lab have spanned. There's been an evolution of being very excited early days in the field about doing therapeutic tissue engineering, as exemplified by this experiment I got involved in as an assistant professor to build the human ear on the back of a mouse. But over the past couple of decades, there's been a real evolution in thinking about the potential of tissue engineering, not just to replace organs in vivo, but to build models of how the human system works so that we can do a better job of looking at safety and efficacy of drugs. And so back in the early 2000s, this idea just wasn't that interesting, especially to students. They came in and they wanted to build a heart that you could transplant or a liver that you could transplant. But there's been a growing appreciation in all communities, particularly because pharmaceutical companies and biotech companies have become more and more stymied about how to reduce the costs of drug development and really look better at safety and efficacy early in the process. There's become a renewed and increasing interest in doing tissue engineering for drug development. So what I want to do today is really a conceptual talk. I'm not going to go into an enormous number of details, but I want to give a flavor about how I came to appreciate the ways that what's going on in the synthetic mammalian synthetic biology community could actually be a very powerful complement to the field of tissue engineering. And I'm going to, again, do it through the context of the lens of drug development. And I know there are a lot of students here, so I'm going to talk about some basic conceptual things in addition to a little bit of data. So this is a schematic of a pipeline, which is arguably not really, this pipeline concept in the drug industry has been evolving, but traditionally you had a disease that you wanted to focus on. We just heard about some cancer therapies in the previous talk, and there have been a number of others at this conference, so maybe diabetes, rheumatoid arthritis, whatever it is, and you find in that disease some kind of target. More and more, they may be cellular therapies, as we heard, but the traditional has been maybe some kind of enzyme or receptor that's involved. You develop a number of compounds, and then among those, you have to pick and take one forward all the way to the clinic. And you pass through. So you have your first generation molecules, you may increase selectivity, potency, you may have some assays for off-target effects, and then you move into a whole series of preclinical studies where you do something called absorption, distribution, metabolism, excretion, ADME, and toxicology. These have traditionally involved animals, but more and more human cell cultures, and then you go into phase 1 and later clinical trials. Okay, and so where we see tissue engineering is able to build, as being relevant here, is taking all the kinds of things you not only do here in classic ADME tox and trying to say, can we predict human toxicology or human how a drug will act in a human system better from that standpoint, but actually starting to move things back into this area here, where we even start thinking about building complex systems that would let us look at efficacy very early in the process, things that might involve the immune system and so on. So this is sort of an overall view of how our lab, in collaboration with a lot of others, are now taking an approach to build systems where we may take samples from patients, build things in vitro, and think about restructuring this pipeline so that we get a lot of data earlier on safety and efficacy in people. I want to give you one example problem. It's one that is very interesting in the drug industry and give you a sense of how you can move through and think about different ways to approach this problem. It's a problem called idiosyncratic liver toxicity, and it happens, and it's a real challenge in drug development because it's a kind of toxicity that's never predicted in the preclinical studies, and not only that, it doesn't show up in the early clinical trials. It only shows up when you get out in very large patient populations. All of a sudden, you have adverse events showing up, sometimes fatal, and the drug has to be pulled after over $1 billion has been invested in it, sometimes more. Now, of course, there are lots of hypotheses about why this happens, and 3 of them are shown here. So it could be due to some genetic dysregulation. And in fact, genome-wide association studies have revealed for some drugs genetic associations. But guess what? You think they'd be in liver drug metabolizing enzymes, right? It's maybe 2D6 or something like that. Turns out they're mainly immune system alleles that show up in these kinds of studies. There could also be adducts that are protein adducts that are made from the drug, and that's known to happen. Things like penicillin and antibiotic allergies come from that. But one that's really becoming more appreciated, and now data are accumulating that it's actually true in the clinic, is that there's some kind of synergy between inflammation in the liver and metabolism of the drug. So metabolic pathways synergizing with inflammation in the liver. Now, where would liver inflammation come from? As you may remember from anatomy, or can learn it right now, your liver and gut are intimately connected. Everything, all the blood that leaves your GI tract goes immediately to the liver and into the portal vein and feeds the liver. And all the time, you've got all these microbes in your gut, and all the time, this minute, no matter what you ate for breakfast, a few little molecules of those microbes shed lipopolysaccharides and things like that, leak through the gut and go to the liver. And they interact with the immune system in the liver. This is actually important. Your gut microbiome plays a huge role in regulating your immune system systemically and certainly in the liver. In fact, mice that don't have microbes in their gut don't develop a normal immune system. So it's a very important interaction that we're really only beginning to understand. But from the standpoint of this problem, anything that increases gut permeability, it could be an infection, a GI infection. It could be the drug itself. And actually, Amgen had a really difficult case where the drug itself increased the gut permeability. Anything that increases gut permeability, even transiently, will allow more lipopolysaccharide to land in the liver. And you can get up to a level that maybe you wouldn't even notice it if you weren't taking the drug. But together, then, with metabolism of the drug, which can stress and deplete glutathione and things, you get this terrible thing where you crash. You get an infiltration of immune cells and so on and so forth. And so right now, you can imagine people might be taking a drug chronically and then, go on a trip. Like I just got back from Brazil, and I'm having probably some of this going on. And so if you're taking a drug, you may have a transient thing that could actually lead to this, especially if you've not been sleeping and things. So one way, you'd like to study this. You could say, well, how could we study this? Could we take we'd like to take this really complicated situation and make it as simple as possible. And so could we replicate it with just taking hepatocytes, which of course in that model are the ones that are getting slaughtered by all of this inflammation drug, and so could we do this where we just take a model of adding cytokines to mimic that inflammation plus our drug, and maybe plus, you could do this for infection and so on. And then ask questions in an in vitro culture, are there rules about how you get this inflammation to interact with drugs that would govern whether the cells live or die? So in fact, in a project with Pfizer, and actually what you'd love to do is furthermore ask, are there rules about how the cells are processing information through signaling pathways in order to respond? Because then maybe you could mediate this kind of toxicity. So in fact, with Doug Laufenberger and Pfizer, and Peter Sorger, we did a study together with Pfizer to ask this because they were very interested and they had a whole lot of drugs. Could you do this by giving a lot of, combining different extracellular cues, some pro-survival, some pro-death, measure the effects of these across multiple different signaling pathways, and look at the responses. So this is a way to take a sort of systems biology approach. In systems biology, you reverse engineer things and try to learn the rules that way. So in fact, you can do this, or you like to do this, because just regular old hepatocyte cultures, drug companies do those right now. And they do all kind of assays and 96 well plates or more. So this is a very attractive way. If you're in the drug company, we've got an assay, we add our cytokines, drugs, and look at this. Maybe we can find out out rules that would let us get the signaling. So in fact, this has all been published, so I'm not going to go through this data in gory detail, but what we did together is took 90 drugs, some of which were known to, all of them had extensive data in people. Some were toxic, some were known non-toxic, so you have pairs, and we did these high-throughput experiments, and we said, do we see death? So basically, this line here is related to a clinical concentration threshold that you'd use, and any drug that's shown in red here in this toxic list, we were able to find synergy between inflammation and toxicity by this simple assay. And over here, not a single one of the really, really well-accepted non-hepatotoxic drugs showed up as a positive. One here, clarithromycin, is viewed ambiguously in the community. Some people actually think it's an idiosyncratic toxin. So this was a great assay, except for the fact that only a fraction of these drugs, known drugs, were captured. So one might reasonably ask, one more thing. And we could go in, actually, in the same experiment, measure signaling pathways and find that there is a synergy between these. You can reduce, with all these pathways measured, it turned out that these 4 pathways were critical in regulating pro-survival and pro-death signaling. But we didn't capture all the drugs. We don't know everything about this toxicity. And we're still thinking about how there may be other things coming from the gut that are not replicated by our simple cytokine mixes. So the question is, do we need more tissue complexity? Okay, so do we need more tissue complexity? And the answer is yes. And then the question is, how do we get it? How complex do we need to be? I don't have the definitive answer to that. I'm going to step you through some ideas, because today is about, I'm here for applications and to really kind of give some ideas. So one thing I want to say, because this is a field we've worked in for a long time, generating these kind of more complex 3D models, one thing I want to say is there's always, always a tension between how complex something is and how easy it is to use. And I'm illustrating that here by a picture of me and my older brother. And I had for a long time, when I was in graduate school at Berkeley, a Kawasaki KZ440, and it worked great. It was a very reliable bike, but I got a yen for a Ducati, and I got a Ducati. I actually won it in a bet. And that thing was so much trouble. It was so finicky, I hardly ever rode it. And part of it's Cambridge, but I hardly ever rode it. Now, my brother has a Honda. He actually now has a Honda CBR 600RR. And so I actually sold this, and now I'm getting a Honda because it just works better. So you always want to use the easiest thing that works for you, okay? And a drug company, and I'm not embarrassed to admit that I want to go back to a smaller Honda. I have no embarrassment about that at all. So the question is, you can make really complex structures, and I've made them in my lab. But when you go into a drug company lab, it's got to be something easy to set up, easy to use, and easy to get data out of. So we've focused on taking a fairly simple approach, and actually in most areas of doing in vitro tissue engineering for drug development, the workhorse kinds of cells that are used are cells that are primary cells isolated from organs that are rejected from transplant or some other means. In liver, we get resected little piece of livers. When there's a tumor taken out, you have a lot of good liver over here, and you can take those cells. And so we, right now, have been taking an approach to assemble those cells. That's probably going to change very soon, but for now, that's really the gold standard, and that's where most of the work in the field has come from. So we took a pretty simple approach here to take a thin scaffold that, and it's not showing up. I think I didn't set my resolution properly here. But this is just showing an older picture of our first steps to this were to take a silicon chip and just bore holes through it to create a little scaffold that could organize dissociated liver cells on a length scale about the size of a capillary bed. And this is just showing, you can see actually liver sinusoidal endothelial cells here. And the idea being that this is something very easy to set up, that you can flow medium through and distribute organs in. Now, there are ways, and I was involved in the very early days of three-dimensional printing, and there are lots of microfabrication methods now. In fact, we're using them in our lab. But this approach, actually, there's some things that make it have some advantages. You can get cells to self-assemble. So the principles that we started this with is going back to some developmental biology ideas where there are some physical self-assembly rules. For example, cells form cadherin bonds. It's actually a slide that we use in the physical chemistry class that we actually teach in this room, trying to just explain how you can look at cells in the same way that you do high surface tension and low surface tension liquids. So there's been proposed by Malcolm Steinberg in the 60s, actually, that you can try to get cells sorted out by looking at the differences in their quantitative differences in the expression of their cadherin receptors. And he had some beautiful work in the field. This was genetically engineering some fibroblasts to express different levels of cadherins, showing then that if you mix those two cell types that have different levels of cadherins over an appropriate length scale and time scale, they'll actually separate out based on adhesion-guided morphogenesis. Okay? And he went even further and proposed that if you had now two cell types in a surface, you could generate even more interesting structures by looking at cell-cell and cell-substrate adhesion. And in fact, we did a little experiment testing this idea for morphogenesis of hepatocytes cultured together with endothelial, simply, again, doing a very simple thing, varying the amount of matrix that's adherent or present on the substrate so that you have a high adhesion environment, a medium adhesion, and a low adhesion, and you can see you can drive morphogenesis of tissue-like structure. So these are some simple concepts that are fairly robust in getting cells to form 3D structures with features like tissue, not exactly the same. But the question is it useful for some of these drug development assays? So again, we just simply developed a simple reactor that had an array of these little holes in a chip and a membrane here. The membrane actually supports the cells right when you seed them. But the cells attach to the walls of the scaffold. Then you initially have the medium flowing this way. You reverse it. The cells stay attached. The purpose of the membrane is to distribute flow. It has a very high hydraulic resistance. And you can actually get fairly reasonable tissue-like structures from this. Moreover, this kind of simple reactor design is very amenable to scaling up in a way that you can use it in a pharmaceutical company. In fact, we've worked with Pfizer and others and taken it into their labs. There's now a company that has commercialized this and adapting it to a multi-well plate format and showing that you can do things like, this is just showing that in all of these systems, perfusion flow is very important. Power goes off in the labs. We've done this many, many times. The pump goes off, and you can see the red cells are dead, the cells die, and you need flow to keep them alive. So you can go and you ask, well, drug companies always want to look at clearance of drugs and people. And so you've got to show that your system can do some of the simple things, straightforward things. So this is simply showing that we can take this kind of system and actually start to predict clearance of a number of model compounds for which we have a lot of human data. But the question then becomes, can we do the more complicated things? Can we start using even this next level of complexity? It's not super complex, but it's the next level of complexity to start getting back at these questions of how an inflammation would influence the liver. And so if we construct our tissue so that it includes not only the hepatocytes, but these other cell types, the endothelial cells, the stellate cells, and importantly, immune cells, we can start to capture more of this toxicity. So this is just an example showing again, this is the tissue, just a little 3D tissue. This is about 0.3 of a millimeter and 200 microns thick. So if you give the drug alone, you can see the cells are just fine. If you give an inflammatory cue that would come from the gut, lipopolysaccharide alone, you get a little bit of death. But now when you give them together, you see this real synergy and inflammation-mediated toxicity. So this is helpful, but you can say, I'm not really comfortable with the way that you just threw that big bolus of lipopolysaccharide in there. Does that really represent what's going on in patients? Don't we need a more nuanced relationship between the gut and the liver? Why not build the whole enterohepatic circulation, because you've got the liver puts things in bile, it goes to the intestine. Your microbes in your intestine can actually take drugs that were transformed in the liver and transform them back, so now they get taken up again. So you can have drugs that cycle around and around. So can we move toward building this? And the answer is, I hope so. You may have noticed on the first slide I had tissue engineering circa 1993 with the ear, and then tissue engineering 2017 with the body on a chip. So in last year, DARPA, in consultation with the FDA, and then NIH also got together, and they launched this huge program. It's about $150 million total right now, program called the Microphysiological Systems Program. And the dream of the folks who are funding this is that we could actually start to build what they call the human body on a chip, but what we call here at MIT more the human physiome on a chip. And the idea was, from the DARPA side, they wanted to have us build a platform that would let you have 10 interacting microphysiological systems. And so that could be, maybe mini organs might be a way to think about it, but it's really, they avoided the word organ because you can even think of the intestine. It's got all these different subregions. You've got the upper GI, the lower GI, et cetera. So you want to model how many of these things interact together. So of course, we think about enterohepatic circulation. And again, the idea is that you build your little systems off platform. You would put them on like a chip that has 12 livers, 12 guts, 12 whatever, grow them up, and then combine them on this platform. And so we got funded at MIT, a team that I lead at MIT, at a little over $30 million over 5 years, to put together a team of people who could both build these organ systems, put them together on chips, and importantly, figure out ways to get information. So we're really trying to get information out of these in a systems biology approach, like I showed earlier, but now bumping up the level of complexity. It's a big team, and I'm only showing this, simply to illustrate there's a lot of things in here that are hardcore nuts and bolts engineering, mainly done at the Draper Laboratory, which is local, a company, and so on. Lots of folks at MIT, including Eric Alm on microbiome. But a real reason I'm showing this slide in the synthetic biology meeting is that part of what they want us to do is interact with people in the community. We're supposed to build a platform and really give it to people who want to build microphysiological systems. Not only companies who want to do drugs, but researchers, like if you go and look at the posters, I saw Patrick Guy and Ron Weiss's lab has a poster on these liver development things from engineered, where you put circuits into cells. So one of our goals is actually to share these with the community. And NIH has been encouraging us to interact with folks they're funding through their parallel program. And I'll just mention, because it's relevant here for the problem I'm talking about, Jim Wells, who's got a lab at Cincinnati that focuses on, he's a developmental biologist, and he has a really neat way of directing differentiation of human iPS cells into forming gut organoid structures. And it's some really beautiful early work, and this is simply a picture from his paper showing that he can get all three layers, the underlying sort of stromal cells, get crypt-like structures. Can't do it completely reproducibly, and that's why they want to interact with the engineers. So we get back to this idea. We've been assembling things, we're moving toward building that. We've been assembling things, we're moving toward directing differentiation. And I'm very excited about that as a way that we interact here with the synthetic biology community. Again, a huge focus, and I think for the future, will be how do we study inflammation? Many of the diseases we want to develop drugs for involve inflammation. And so we know that crosstalk between organs involves inflammatory, a lot of it involves not only hormones, but inflammatory cues. So a very substantial part of our program hinges on building not only these in vitro systems, but trying to put together the ways that we measure information across all the different kind of omics that will let us get this out. Again, reverse engineering the system to understand it with an eye not only toward working with pharma companies in their drug development process, but ourselves helping them frame questions. And so I'll end just on this last slide. One of the things we are looking to do with these is really help understand diseases. Let me actually go through all the animations. Disease etiology, and a major focus in our team is women's reproductive health and a disease called endometriosis, which is characterized by uterine lining growing outside the uterus where it shouldn't be, in the abdominal cavity, all over these places. There's roughly, I think, maybe 200 and something people in the room. If 50% of them are women, 100, at least 10 women in this room have this disease. And you may or may not have had it diagnosed. It is really, really hard to diagnose because it requires surgery. We don't understand what causes it. They're not very good drugs, and we can't diagnose it except for surgery. So a thing we're very interested in is and this is showing a lesion on the small bowel. This patient probably has a lot of GI problems, maybe increased intestinal permeability. But we're trying to understand why patients present with a whole range of symptoms. So for example, the disease, though not cancer, can actually invade the bowel wall, not just have a superficial lesion. This is a patient, one of the best surgeons in the world for looking at invasive diseases in Brazil, and I actually just got back from visiting him. He took out 10 centimeters of this patient's bowel because she had these two huge tumor-like lesions sitting in the wall that were invasive disease. So one of the things we're trying to understand is, how can you stratify patients by taking some of their samples and bringing them back to the lab and putting them in these kind of in vitro systems. And this is just showing it goes to the lymph nodes. And so this is my last slide, and I kind of put that last bit there because I want to make one point, and it has to do with why I got involved with Ron's vision of this center. In systems biology, which we've been involved with a lot in trying to reverse engineer these signal transduction pathways and how tissues assemble and so on, we've made a lot of progress, but there's whole other ways that information flows in cells. And you can reverse engineer by kind of, knocking out different parts of the information transmitting system, but the opportunity in my field for synthetic biology, in my view, is forward engineering that, combined with the reverse engineering, actually lets you understand and control information flow in these systems much better. So it's really, you learn things by forward engineering you had no idea were happening in cells. And so that, to me, is the excitement that combining these will get much further in both building our tissue engineering systems, but also in helping us understand what's happening in complex diseases like I discussed. That's it.
Questions?
That was a great talk, Linda. Thank you. So I'm really glad that you mentioned the problem of reproducibility of some of these complex systems. And I'm wondering, given that parameter space is so huge, and there's maybe different sources of cells, and do you have an idea of what kind of approaches should be taken, or what might be the sources of some of that irreproducibility of putting together these complex systems and having them respond in the same way twice?
So the question has to do with reproducibility of complex systems, and there are multiple different ways that systems could be variable. One is if you had exactly the same source of cells and you construct things that are different. Okay? And that happens. And that's what we try to focus on, having a simple way to construct things so you get some reproducibility. Another, of course, is the source of cells. And this is a great debate in the community about how cells from, even for right now, when we assemble things, different donors will influence things. So this is part of why we think about systems biology, because there are ways to probe systems that let you sort of smooth out some of that variability by driving the system into different states. And I gave that just high-level view of it in that idiosyncratic tox hepatocellular culture, because we actually did use cells from different donors, and you could have some common signal transduction themes emerge out of it if you put your probes in the correct way. So what I can say, there is variability. I think we strive to have the least amount of variability in what we make. We're inherently going to, likely for a while at least, have variability in the cell sources. This may be solved by having iPS cells. Right now with hepatocytes, we're still a long way from doing the kind of bread and butter things I talked about here, although there are emerging some applications in disease modeling. So that remains a huge challenge because there's also expense and other things associated with it. So I think these are problems that by combining sort of the information side together with the experimental side are going to be solved sooner rather than just focusing on the experimental side.
So what is the status on developing these mature liver systems from ES cells or iPS cells? Because that seems to be the key issue to move it forward, to look at genetic components which are important for what you read out. So my understanding is you can't get really mature hepatocytes from ES cells or from iPS cells.
Wait, sorry, what?
Mature hepatocytes for your system from ES cells or iPS cells.
Right. So I think, again, there's a whole spectrum of things that you want to do in drug development. Some of them involve looking at genetic variation explicitly, or where you know there may be genetic variation. Certainly with diseases, in some cases, there are known problems with certain metabolizing enzymes like 2D6 and so on. So in that case, the ability to generate iPS cells, even to look at metabolites and how a particular, usually a small molecule drug would be metabolized by the whole processing system in the cell, is very useful. In other cases, I think there is a growing appreciation that we have this huge component of the immune system and secretion into bile. So what I'm interested in learning more about is whether you can develop a 3D system. We're getting to the point that you certainly see the starting of systems from ES cells. How do we get the other cell types that are present in liver, because the liver is a huge immune organ, and how do we get the whole reticuloendothelial system to grow together with the hepatocytes? Because hepatocytes are important, but these other cell types and many of the things that we care about, many of the drugs that come out, antibodies that modulate the immune system, are interacting with other cells besides hepatocytes to cause either efficacy issues or toxicity issues. So this, to me, is a huge challenge. We can get the hepatocytes, and maybe for a while we combine them with other cells.
Okay, one last question. Zev?
Just speak a little bit about, so you spoke a little about the tension between complexity and simplicity of some of these systems, but you also speak about the tension between mouse models and human in vitro models for testing some of these diseases. How you go one way or the other?
So I hardly know anybody I interact with a lot of pharma companies. Nobody really wants to do things in mice for a lot of these things. It's just the difference in species, and we actually looked at that in our in vitro, like that initial hepatotoxicity inflammation model. Even HepG2 cells, which are a really terrible model of human liver and drug stuff in vitro, were better than primary rat cells for capturing some of this human kind of where there was known human data. And so I think in terms of the mouse, there are, again, I'm not going to speak all of this. Mouse gets beat up a lot lately because it doesn't do all these things that humans do. I would argue, human in vitro systems also don't do a lot of things you'd want. So there are, for example, you can humanize a mouse liver, and that's being used to test certain hepatitis therapies and so on. So there are some